All right, so again, switching gears big time. We're going to get into population and evolutionary genetics. So we really are kind of going back to kind of our Mendelian genetics that we did in the very first unit, so all the way back, first and second unit for this class. So we are moving out of the realm of molecular biology and up to population level. So we're really like not even looking at organisms anymore. We're jumping all the way up to allele frequencies in an entire population, which is why I feel like it's kind of easier. So we were just talking about mutations and genetic variation. Notes. So there's just a couple of examples of variation here. You can look at variation in a population at all different levels. So you can look at variation in the gene sequence. You can look at variation in the phenotypic effect. And then you can look at variation from the population level. So we can look at molecular, we can look at individual, and then we can look at population. So we're kind of jumping back to evolution here. We're going to kind of be speaking about where genetics and evolution overlap. When I was an undergrad, I had all the biology classes, whatever I was required. And then one of them was evolution and ecology. And I hated that class. I absolutely hated that class. I don't know what was wrong with me, because now when I have to teach ecology, I'm like, oh, I love this. It's like the National Geographic Channel. I love it. It's amazing. But I think I didn't like it because I didn't understand. I was doing cell biology, molecular biology, biochemistry, all of that business. And I didn't understand what evolution had to do with anything. And I kind of understood like the ecology part of it and like, yay, this is interesting, predator, prey, all of that. But I didn't understand what ecology and evolution had to do with everything else that I was studying. And it wasn't actually until I started teaching, and I had to teach Hardy-Weinberg, that I was like, oh, I get it. They're actually related, but nobody ever explained that to me, or they did, and I brain dumped it the minute they said it. That's possible too, because that happens when you're a student. I think you don't have to admit that, but it happens. So I actually really hated when I had to take population genetics and evolution, because I did not understand the relationship between it. But genetics is basically directly leads us into evolution when we are looking at the population level. And of course, whatever's going on in the population is also encoded back down at the molecular level. So you are all probably familiar with the idea, the definition of a population. A population is basically just a group of interbreeding organisms. We are concerned in this class with the genetics of the population. So as long as they are interbreeding and passing on their genetic information, we are interested in this group of individuals. When we are looking at population genetics, we're really not talking about asexually reproducing organisms, because we are looking at how genomes combine and how allele frequencies change in a population. Part of the population that, as a genetics class, we are most interested in is the gene pool, which is just the whole collection of genes in a population. But we are going to be looking at those genes one at a time. So we're going to look at how allele frequencies of a single gene change or fluctuate in a population. Okay, so variation can exist at the phenotype level. So we can have sequence differences. So we can talk about how the genetic sequence changes in the population. And we can also look at how the sequence or the change at the molecular level affects the overall phenotype in our population. But they are related. So if evolution is working on a phenotype, causing a phenotype to change frequency in a population to become more or less common, then the sequence is also going to become more or less common. And so one of our requirements for evolution is that evolution only works on traits that are going to be controlled, or we should maybe say coded, at the sequence level. In other words, if there's no DNA sequence or gene that is related to a particular phenotype, then that is not going to be passed down to the next generation. And so evolutionary forces are acting on a phenotype directly, but acting on the gene pool of the population indirectly. And so what this means is that if a particular phenotype survives, and in 101 we would have talked about the different mechanisms for evolution. We'll get to that in a little bit. But if a particular phenotype survives and is passed on, then the genes are also passed on. And so we're going to change our gene frequency based on who's passing on their alleles. Did you have a question, DJ? I just saw your hand move. All right. So to define evolution, evolution is a change in allele frequencies in a population. So when we say allele frequencies, we are talking about the proportion of alleles in a population for one specific gene. So evolution can be working on multiple genes, multiple alleles at the same time, but we are going to isolate it to just one. We're going to be looking at how one allele changes within a population. So when you're studying evolution, and this is what I didn't get when I was an undergrad, I didn't get that when you're studying evolution, you're really studying the genetics of a population and how the genetics are changing. So evolution can kind of be divided into sort of two big branches. We've got microevolution. In microevolution, we are specifically looking at the changes in allele frequencies. And so that is going to be our focus for this class. The other side of evolution is what we would call macroevolution. So macroevolution is big, broad patterns of evolution, origins of new species, drastic changes, origins of new traits, etc. So mostly we're going to focus on microevolution, changes that are happening at the population or species level. But microevolution can lead to macroevolution. It doesn't have to. Microevolution can just change alleles within a population. It can be minor changes. But if the changes become great enough, we can end up with macroevolution. So when this happens, when we sort of switch between micro, and macro, we get the divergence of one group into two different species. So at some point, if microevolution continues, we can actually end up with two groups that are so genetically different that they can't interbreed and therefore produce new species. We are not going to get there in this class. We're not going to get to new species. We're going to stay at population level and just talk about how populations change in a, as far as their genetic makeup. All right, so the basis of our population genetics is going to be in the genotype frequencies and the allele frequencies in a population. So we calculate both of them the same way. A frequency is just generally going to be the number in a subset divided by the total. So we just have to keep track in this chapter for if we are calculating genotype frequencies or allele frequencies. So when we were learning about Mendelian genetics, we learned that all individuals have two alleles for any particular gene. And you can be heterozygous or homozygous for those alleles. So if we are looking at genotype frequency, we are looking at the combination of two alleles. So out of this group here, if we're looking at the frequency of homozygous dominant individuals, it's just going to be the number of homozygous individuals divided by the total number of individuals in the population. So I will use this notation. This just means the frequency of those homozygous individuals. So that little notation with the F in the parentheses, that's just shorthand for frequency of. So we can calculate allele frequencies in the same way, except this time we're looking at the individual alleles, not the allele combinations. So if you remember using Hardy-Weinberg in Biology 101, we're going to use the exact same thing. There's really nothing that's really new or earth-shattering in this chapter. But one problem that students have is mixing up allele frequencies and genotype frequencies, especially when they have to interpret genetics problem. Hardy-Weinberg problems. So if we need our allele frequency, we're going to figure it out the exact same way. The number of copies of the allele divided by total number of copies in a population. So we might write frequency of dominant allele A is the number of copies of allele A divided by the total. So keep in mind for this one that each individual has two copies of every allele. So if you are asked to calculate an allele frequency, you might have to determine how many alleles are in that population. And that's just going to be 2 times n. N is a number in our population or the population. So depending on what type of information you are given, you may or may not have allele frequencies and genotype frequencies at the same time. So that's where our Hardy-Weinberg equation is going to come in. Before we get there, though, we're going to talk a little bit more about allele frequencies. So we're preparing for Hardy-Weinberg. The general sort of rule for Hardy-Weinberg is that the lowercase p variable is going to represent the dominant allele. And the lowercase q is going to represent the recessive allele. However, it is not a requirement that we only do this with dominant and recessive traits. We can also do this with codominant or incomplete dominant traits. And so usually we're going to assign P the dominant, but we can also just assign it to either one of the alleles. I am going to ask you to please always, if you know which one is the dominant, please always assign it to P. If you know which one is the recessive, always assign it to Q. And that's really just kind of by convention, so I can follow what you're doing. That's just kind of standard. But honestly, it doesn't actually matter which one you apply it to. But this is how you would have learned it in 101, so we're going to just keep it like that. If we need to calculate the frequency of one allele, we have to keep in mind that our heterozygous individuals have only one copy and our homozygous individuals have two copies. So I know that you all know that. We haven't got this far and you haven't figured that one out yet. So I know that you know that. But we can use this sort of general formula to help us calculate our allele frequencies when we know something about the genotypes of our individuals. So if we want to calculate the frequency of that dominant allele, or if we want to calculate P, we can use this generic formula. We use the exact same formula to calculate the frequency of the recessive. So to calculate our total number of total number of alleles, we can take our number of homozygous dominant individuals, and we know that every individual that is homozygous has two copies of that allele. So we can take 2 times the number of individuals that are homozygous dominant, and then we can add that to the number of individuals that are heterozygous, because these individuals only have one allele each. And then for our population size, because we're looking at alleles, we know that each individual has 2 alleles. So we have to multiply our population by 2. The alternative to that, just to confuse you, our textbook does the opposite. And you can also figure it out by taking the number of individuals that are homozygous plus half the heterozygous individuals. I don't care which one you use. Do whichever one makes more sense to you. I feel like this one, the first one, makes a little more sense to me. So that's usually the one that I will default to, because you're taking 2 alleles multiplying by 2. So it just seems a little more intuitive to me. Y'all have any questions on this equation? This is probably new for you. I know I didn't do it for 101. I don't think Charles does this one either, but you had Charles, right?
Yeah.
What?
It's in 102.
I can't remember if we did Hardy-Weinberg in 102. Did we?
I'll dip into it.
In 102. I took Hardy-Weinberg out of 102 this semester. It might have been. I don't remember. It just, yeah. I took it out.
Okay.
That was my contribution to the biology department, is I took Hardy-Weinberg out of 102, which I think made some people raise their eyebrows, but they're not teaching it. Okay. So the usefulness of that formula, that generic formula, is that you can apply it to multiple alleles. So if you have more than one allele for a particular gene, you can do the exact same thing. So for example, if we have a allele 1 homozygous individual, we can take that same formula, add it to our individuals that have the other 2 heterozygous combinations, and still figure out our allele frequencies. So we are really not going to get too much more complicated. We're not going to do more than 3 alleles, but you could do as many as you needed. Just applying the same principle of all of your heterozygous have only half the number of alleles as individuals in the population. So let's go ahead and do a simple practice problem. And then when everyone comes back next week, we'll start with the hard one. All right. So I would like you to calculate the frequency of allele S1 and the frequency of allele S2. We'll go ahead and set S1 for P and S2 for Q, just so we're all doing the same thing. And I wasn't sure if we were going to get to this yet, so I haven't worked this one out all by myself. So we'll do this together. You may use either version of that equation that I gave you, the 2 times the homozygous or half of the heterozygous. Either one, whichever you prefer, is Did you get it? You did that right. And it's 176. That's correct. Total in the population.
179. Oh, wait, that's me.
Okay. I'll say we've got all even numbers there, so we shouldn't have a 9.
Yeah.
176?
Mhm.
Okay.
I pressed.
Okay, so the first thing we need to do is figure out our total population size. So total population is all of the individuals that are homozygous and heterozygous. So for our p-value, we're going to have 2 times the number of homozygous plus the number of heterozygous divided by 176. So what does our p-value come out to be?
About 10.2
It should be a decimal.
decimal.
So we've got 8 plus 38 is 46 divided by 176.
Yes, 26%. 0.26.
0.26.
Yeah.
Or, wait,
And
I did it.
so I got 0.13.
Okay, so on the bottom, what was on the bottom, DJ?
352.
352. I hate doing math on the board. Okay, 352. What does it come out to be?
0.13.
Yeah. 0.13. All right, so our p-value is 0.13. What that means is the frequency in the population is 0.13. And as far as the actual population itself, it means that 13% of the alleles are S1. Down here on the bottom then, you could just tell me what you got, since me doing math up here is clearly disastrous. 2 times 134 plus 38 divided by 356.
About 0.86.
0.86. So that means that 86% of the alleles of the population are the S2 alleles. Just sort of precursor, your total number of alleles is always going to be the frequency of one plus the frequency of the other. So you all probably remember this from Biology 101. Your allele frequencies and your genotype frequencies should always be written as a decimal, because the total can never be more than 1. So if it asks for a percentage, then definitely convert it to a percentage, but otherwise just go ahead and leave the frequencies as it is.